Welcome back all dear students. I am Chetan Digambar Pawar, working as an assistant professor and head department of botany at Gokhale Education Society's Arts, Commerce and Science College, Jawar, district Palghar. Students, today we are going to study the topic from TYBSc Botany paper second, that is a plant diversity 4, and this topic it is included in the unit 3 anatomy. The topic of our presentation is the anomalous secondary growth in Dracaena. Dracaena, it is a monocot. In previous lectures or presentations, we have seen the anomalous secondary growth in dicot stem. Now this is the anomalous secondary growth in monocot stem, that is Dracaena. Now before going to the secondary structure of monocot stem, it is important to overlook the primary structure of monocot stem. That's why first we will study the primary structure of monocot stem. As you can see in the diagram, the monocot stem it is a roughly circular in its outline, and the outermost layer of the stem is the epidermis, which is made up of the parenchymatous cells, and on the outer side it shows a layer of cuticle and few stomata. Then below epidermis there is the multi-layered sclerenchymatous hypodermis is present, which show the patches of chlorenchyma, which is a photosynthetic tissue. Then inside the hypodermis there is a very large tissue is there, which is called as a ground tissue, and this ground tissue it occupies the maximum area on the monocot stem. This ground tissue it is made up of the large parenchyma cells, which are very loosely packed and showing the intercellular spaces. Then within this ground tissue, the vascular bundles they are embedded, and these vascular bundles they constitute the vascular tissue of the monocot stem. These vascular bundles they are numerous in their number, and they are scattered in the ground tissue. These vascular bundles they are of conjoined, collateral and closed type. Conjoined means when the xylem and phloem both are present in a same bundle. Collateral means they are placed side by side, and closed type means the cambium is absent in the vascular bundle. Then these vascular bundles they are again of endarch type. Endarch means when the protoxylem is present towards the inner side and metaxylem is present towards the outer side of the vascular bundle, then that vascular bundle is said to have the endarch type of the xylem. This is a typical type of the vascular bundle which is mostly present in the monocot stem. The outer vascular bundles they are smaller in size and larger in their number, while the inner vascular bundles they are of larger size and they are few in their number. The most important point regarding the monocot stem is that the monocot stem in monocot stem the secondary growth is usually absent. Now moving to our actual topic, that is the anomalous secondary growth in Dracaena stem. Dracaena it is a monocot which shows the arborescent habit. That means wood formation takes place in the stem of the Dracaena. Dracaena belongs to the family Liliaceae. The young stem shows the primary growth with the conjoined, concentric and closed type of the scattered vascular bundles in the ground tissue. The point to be noted that the vascular bundles they are conjoined, concentric and closed type. The concentric type of vascular bundle this is not typically observed in the monocot stem, but in this monocot the concentric vascular bundles are present. Concentric means when there is the absence of width, and directly from the center of the vascular bundle the vascular tissues are present. These vascular bundles, they are of amphi-vessel type or leptocentric type. Amphi-vessel or leptocentric means when the phloem is surrounded by the xylem element, then it is called as a amphi-vessel or leptocentric type, which is having the endarch type of the xylem. Endarch means protoxylem is present towards the inner side while the metaxylem is present towards the outer side. During secondary growth, the parenchymatous cells which are present at the periphery of the ground tissue, they become meristematic and they form an anomalous ring of the secondary cambium. That means the formation of cambium, secondary cambium, takes place from the parenchymatous cells which are present at the periphery of the ground tissue. This secondary cambium ring, it shows the anomalous activity and it cuts off the entire concentric vascular bundles towards the inner side. This is the anomalous secondary activity and it cuts off entire concentric vascular bundles towards the inner side. In this diagram, you can see here, these are the primary vascular bundles which are present in this ground tissue. Then the secondary vascular bundles, they are produced by this cambium. This is a layer of cambium which produces these secondary, these are the secondary vascular bundles which are of concentric type, and these secondary vascular bundles, they are produced by this cambium. This cambium, it produces the secondary vascular bundles towards the inner side. Okay, and these are of concentric and amphi-vessel or leptocentric type. Okay, these secondary vascular bundles, they are small and they are crowded towards the periphery, as you can see in the diagram. They are embedded in the conjunctive tissue. This is the another one important feature, is that in this diagram, the there is the conjunctive tissue is present instead of ground tissue. In primary growth, the vascular bundles are surrounded by the ground tissue, while in secondary growth they are surrounded by this conjunctive tissue. Okay, the cambium produces one or two layered parenchyma towards the outside as well. In this diagram, you can see here, there is a layer of parenchyma is formed outside the cambium. Here you can see that there is a layer of parenchyma is formed, which is formed outside the cambium. Thus, in Dracaena stem, the origin of cambium and the activity of the cambium is anomalous. The important features, we can enlist the important features of the Dracaena stem. The first important feature is that there is the presence of concentric amphi-vessel closed vascular bundles in the ground tissue. The origin of cambium is anomalous because it forms or it develops from the parenchyma cells which are present towards the periphery of the ground tissue. Then the activity of cambium is also anomalous because instead of producing the secondary vascular or secondary tissues intracambially and extracambially, it produces the entire concentric amphi-vessel type of the vascular bundles towards the inner side only, and towards outer side there is a one or two layered parenchyma is produced. Then the development of entire concentric, same point is there, the development of entire concentric secondary vascular bundles towards the inner side of the cambium, and the overall the anomalous secondary growth in Dracaena stem, it is of adaptive type because it helps in the woody structure or arborescent habit of the Dracaena stem. Thank you.